There are creatures out there dwelling in the hidden corners of our world that don't just survive, they invade, control, and ultimately puppet their hosts. This isn't the realm of science fiction, a tale spun for unsettling entertainment. No, this is the stark, often brutal reality of nature. We are about to embark on a journey into a world where the lines between predator and prey, between free will and manipulation, become disturbingly blurred. A world of parasites that can transform an industrious ant into a suicidal zombie, or, more chillingly, make a cautious rat seemingly fall in love with its most feared predator. These microscopic manipulators, refined by millennia of evolution, have perfected the art of biological espionage. They are the unseen forces, the silent puppet masters pulling the strings of life in ways we are only just beginning to comprehend. Their strategies are diverse, their targets varied, but their objective is singular: to ensure their own propagation, often at the extraordinary and sometimes fatal expense of their hosts. The intricate dance of life and death takes on a new, more sinister dimension when one considers these entities. And as we delve deeper into these mind-bending phenomena, a question, unsettling yet unavoidable, begins to surface. If these parasites can exert such profound influence over other creatures, shaping their behaviors, their instincts, even their perceived desires, could they perhaps even influence you? Join me as we explore the astonishing and sometimes terrifying world of nature's puppet masters. In the humid, teeming rainforests of the world, a drama of extraordinary manipulation unfolds daily, largely unseen by human eyes. Here, amongst the decaying leaf litter and the vibrant green canopy, lives an ant, a creature of remarkable social structure and industry. But even this tiny titan of the forest floor is not immune to the insidious strategies of a particularly cunning parasite. I speak, of course, of the fungus, more chillingly known as the zombie ant fungus. Its life cycle is a testament to the brutal efficiency of natural selection, a macabre ballet directed by a fungal intelligence. The process begins with a single microscopic spore. This unassuming particle, perhaps wafted on a gentle breeze or brushed against by a foraging ant, is the harbinger of a profound transformation. Upon landing on an unsuspecting carpenter ant, the spore germinates, its hyphae tiny thread-like filaments penetrating the ant's tough exoskeleton. This initial breach is subtle, almost imperceptible. The ant continues its duties, unaware that an invader has begun to weave its network throughout its body, a silent, growing presence that will soon commandeer its very being. As the fungus proliferates within the ant's hemicole, its body cavity, it begins to release a cocktail of chemicals. These are not random byproducts, but precisely targeted compounds designed to alter the ant's behavior in very specific ways. The once social creature starts to exhibit erratic movements. It may stumble or become disoriented, straying from the colony's well-trodden paths. It is as if an invisible hand is now guiding its actions, overriding the ingrained instincts that have governed its life until this fateful infection. The fungus remarkably does not seem to invade the ant's brain directly, at least not initially in a way that destroys it. Instead, it appears to control the muscles, turning the ant into a living vehicle, a biological machine subservient to the fungus's ultimate goal. The ant is driven, compelled by an irresistible urge to abandon its colony, its lifelong companions, and embark on a solitary final journey. This journey is not random, it is a carefully orchestrated ascent. We observe the infected ant, now a mere puppet, climbing the stem of a plant. It moves with a strange, almost preternatural determination, scaling heights it would not normally venture to. The fungus is guiding it to a very specific location, typically a leaf on the north side of a plant, around 25 centimeters above the ground. This precise positioning is crucial, for it provides the ideal conditions of temperature and humidity for the fungus's next and most critical stage of development. Once the designated altitude and orientation are achieved, the ant performs its final gruesome act of servitude. It bites down, hard, into the main vein of a leaf, its mandibles locking in a death grip so powerful that the ant will remain fixed even after death. This act, driven by the fungal puppeteer, anchors the ant, transforming its body into a stable platform from which the fungus can complete its life cycle. The ant itself is now dying, its internal tissues largely consumed. From the back of the deceased ant's head, a fungal stalk, the fruiting body, begins to emerge. Our journey into the world of parasitic manipulation now takes us from the humid rainforest floor to a far more familiar setting, perhaps even into our own homes. Here we encounter a single-celled protozoan, a parasite of remarkable cunning and widespread distribution. Its life cycle is a complex narrative involving, crucially, felines as its definitive host, and a range of warm-blooded animals, including rodents and indeed humans, as intermediate hosts. And it is within these intermediate hosts that performs its most astonishing feats of behavioral engineering. The primary stage for reproductive ambitions is the gut of a cat, be it a wild leopard or a domestic tabby. Here the parasite reproduces sexually, producing resilient oocysts, which are essentially egg-like structures. These oocysts are then shed in the cat's feces, potentially millions at a time, into the environment. They are hardy survivors, capable of remaining infectious in soil or water for many months, awaiting their chance to encounter an unsuspecting intermediate host. Consider a mouse or a rat foraging for food. These creatures possess an innate, deeply ingrained aversion to the scent of cats, a vital survival mechanism honed over countless generations of predator-prey interaction. The smell of cat urine, in particular, triggers an immediate fear response, prompting the rodent to flee. This is where stages its most audacious intervention. If a rodent ingests oocysts, perhaps from contaminated soil or water, the parasite springs into action. Once inside the rodent, the oocysts release tachyzoids, a rapidly multiplying form of the parasite, which disseminate throughout the body. Eventually, they form tissue cysts, most notably and significantly within the brain, and it is here, within the neural pathways of its host, that begins to subtly, yet profoundly, rewrite the rodent's behavioral script. The parasite doesn't merely make the rodent ill, it changes its very perception of danger. The most striking alteration is the loss of that crucial fear of cat odors. Infected rodents no longer scurry away from areas marked by feline urine. In fact, some studies suggest something even more bizarre. These infected rodents may even develop a fatal attraction, a perverse curiosity towards the scent of their mortal enemy. It's as if the parasite has flipped a switch in the rodent's brain, transforming a life-preserving fear into a dangerous, almost suicidal lure. This behavioral shift is, of course, entirely to the parasite's advantage. The natural world is replete with such tales of parasitic puppetry, each more inventive and unsettling than the last, and are but two players in a vast hidden orchestra of manipulation. Let us now turn our gaze to other masters of this dark art, creatures that have evolved equally astonishing methods to bend their hosts to their will, showcasing the sheer diversity of these mind-altering strategies across the animal kingdom. Consider, for instance, the lancet liver fluke. This tiny flatworm embarks on a complex life cycle that involves commandeering not one, but two intermediate hosts, before it can reach its final destination in the liver of a grazing mammal, like a cow or a sheep. Its first unwitting accomplice is a land snail. The snail consumes slime balls containing the fluke's cercaria, shed by ants. Inside the snail, the parasite develops and is then excreted in the snail's slime. This slime, laden with parasitic larvae, is then consumed by an ant, and here, the truly remarkable manipulation begins. Most of the fluke larvae insist within the ant's abdomen, but one, or sometimes a few, undertakes a perilous journey to the ant's subesophageal ganglion, a cluster of nerve cells that functions somewhat like a brain. Once there, this brainworm takes control. As evening approaches and temperatures drop, the infected ant, instead of returning to the safety of its nest, is compelled to climb to the top of a blade of grass. There, it clamps its mandibles onto the grass tip and waits, immobile, throughout the cool night and into the early morning. This bizarre, summit-seeking behavior places the ant directly in the path of grazing herbivores. If the ant is not eaten by dawn, and the temperature rises, the fluke temporarily relinquishes control, allowing the ant to behave normally, only to reassert its command as the next evening falls. This precise, temperature-sensitive manipulation dramatically increases the chances of the ant, and thus the fluke, being ingested by a suitable final host. Then, there is the ghoulish spectacle orchestrated by the parasitic hairworm. These incredibly long, slender worms develop inside terrestrial insects like crickets and grasshoppers. The adult worms, however, are aquatic and must return to water to reproduce. But how does a worm, trapped inside a land-dwelling insect, achieve this? Through a chilling act of behavioral modification. As the hairworm reaches maturity, it induces an irresistible urge in its host to seek out water. The cricket, which normally avoids water, will now actively search for it. A pond, a stream, even a swimming pool, and driven by this parasitic compulsion, will leap in. Once the host is submerged, the mature hairworm, often many times the length of its host, dramatically exits the drowning insect's body, writhing free to find a mate and continue its life cycle. The unfortunate cricket, its purpose served, is left to perish, a discarded vessel in this parasitic drama. The sight of a hairworm emerging is truly one of nature's more unsettling spectacles. In the marine realm, we encounter the parasitic barnacle, which inflicts a particularly insidious form of manipulation upon crabs. A female larva, upon finding a suitable crab host, injects a small cluster of cells into its body, typically at a joint in one of its legs. These cells migrate through the crab's bloodstream, eventually settling in its underside where they grow into a structure called the externa. Visible as a fleshy sac where the crab would normally carry its own eggs. But the influence extends far deeper than this external growth. It effectively castrates the crab, preventing it from reproducing. More astonishingly, if the host is a male crab, the parasite feminizes it. The male crab's abdomen broadens, resembling that of a female, and it will even begin to perform female grooming behaviors, meticulously caring for the externa as if it were its own precious brood of eggs. It will fan and clean the parasitic sac, unwittingly aiding the dispersal of the next generation of larvae. Each of these examples, from the fluke-driven ant to the zombified cockroach, paints a picture of a natural world far stranger and more complex than we often imagine. Having journeyed through these remarkable and often unsettling examples of parasitic mind control in the animal kingdom, we arrive at a question that inevitably looms large. Could humans be similarly susceptible? Are we, with our complex brains and sophisticated consciousness, immune to such insidious manipulations? Or could our own behaviors and thoughts be subtly shaped by the microscopic passengers we carry within us? This is a frontier of scientific inquiry that is both fascinating and, for some, deeply disconcerting. We have already touched upon a parasite that successfully infects a significant portion of the global human population and forms dormant cysts in our brains. While the dramatic behavioral changes seen in rodents, that fatal attraction to cats, are not replicated in humans, the research into more subtle psychological or behavioral correlates is ongoing. Some studies have tentatively linked latent toxoplasmosis with alterations in personality traits such as increased risk-taking, impulsivity, or even subtle shifts in reaction times. It is crucial to approach these findings with scientific caution. The human brain is an organ of immense complexity, and behavior is multifactorial, influenced by genetics, environment, experience, and social context. Attributing specific behavioral changes solely to a dormant parasite is a challenging task, and much of the current research points to correlations rather than definitive causation. Yet the very possibility that a single-celled organism could subtly nudge human behavior remains a compelling hypothesis, one that underscores our biological nature. Consider the burgeoning field of research into the gut-brain axis. We now understand that the vast ecosystem of microorganisms residing in our digestive tracts, Our gut microbiome communicates extensively with our brains, influencing mood, stress responses, and even cognitive functions. While most of these microbes are bacteria, and many are beneficial or commensal, the gut can also be a habitat for various parasites. Could these two leverage this intricate communication pathway to their advantage? The mechanisms by which gut microbes influence the brain are complex, involving neural, endocrine, and immune pathways. They can produce neurotransmitters or their precursors, modulate inflammation, and affect stress hormone levels. If beneficial bacteria can have these effects, it is plausible that certain parasitic organisms, co-evolving with us over millennia, might have developed strategies to subtly manipulate these systems to enhance their own survival or transmission, perhaps by altering host appetite, social behavior, or even mood states in ways that, however indirectly, serve the parasite's life cycle. The concept of the extended phenotype, popularized by Richard Dawkins, is particularly relevant here. It posits that a gene's effects are not necessarily limited to the body of the organism in which it resides. A parasite's genes, therefore, can express themselves through the altered behavior of its host, if that behavior benefits the parasite's propagation. The zombie ant's death grip, or the cricket's suicidal leap, are classic examples of a parasite's extended phenotype. Mind control, as we have seen, is not merely the stuff of horror films or speculative fiction. It is a tangible, pervasive force in the natural world, enacted with astonishing precision by organisms that have honed their manipulative skills over eons. It's happening all around us, in the rustling leaves of the forest, in the depths of the ocean, and perhaps in ways we are only beginning to grasp, even inside us. The intricate dance between host and parasite reveals a layer of biological reality where autonomy can be a fragile illusion. In the grand, often brutal theater of nature, survival isn't always about sheer strength or speed or overt aggression. Sometimes it's about control, the subtle, insidious ability to influence, to direct, to commandeer the will of another. And the puppet masters, these architects of behavioral change, they are often smaller than we can easily see, their influence woven into the very fabric of life. Are you sure your thoughts are your own? STORYSTATIA, the truth will disturb you.